Welcome to the BioInsights, where science comes alive.
Today, we're going to completely change how you look at industrial fermentation. I want you to picture a modern fermenter not just as some giant steel vat of bubbling liquid, but as a highly controlled, high-tech microbial factory. Inside this factory, we've got literally billions of microscopic workers, the microbes, manufacturing incredibly valuable products for us. We're talking everything from life-saving antibiotics in your medicine cabinet to essential food ingredients you eat every single day. But here's the catch. These workers are super fragile. They're constantly altering their own environment, and if we don't meticulously control their workspace, the whole factory crashes. So let's dive right in and see exactly how we keep this microscopic workforce alive and productive.
To run this critical systems check, we're going to master 5 main control panels today. First, battling the acidity problem. Second, measuring microbial breath. Third, the dissolved oxygen probes. Fourth, the power of positive pressure. And finally, the master control room.
All right, let's start with system check number 1, battling the acidity problem with the pH control panel.
The biggest, most immediate danger to our microbial workers is actually themselves. The thing is, these microbes are incredibly messy. As they gobble up nutrients and pump out the metabolites we want, they naturally generate acid as a waste product. If we just leave that unchecked, the environment inside the tank becomes toxic really fast. Think of it kind of like maintaining the pH of a massive swimming pool. If you aren't constantly monitoring and adjusting the chemicals, the water quickly turns cloudy, green, and completely unsafe. Well, in a fermenter, the stakes are literally a matter of life and death for the yield. If the pH drops too low, those microbes will acidify themselves to death, and production just grinds to a halt. So constant monitoring is absolutely vital here.
Now, our strategy to fight this acidity has completely evolved over the years. Back in the day, scientists had to rely on manual adjustment. They'd use natural buffers like phosphates or calcium carbonate, or, and this sounds exhausting, they'd have to physically pull samples from the bubbling broth, test them, and manually dump in acid or base. It was so laborious, and the data was always delayed. You were constantly reacting to the past. Today, though, we use sterilizable probes that sit directly inside the fermenter. These things are automatic and continuous. The absolute second they detect a spike in acidity, they automatically trigger the injection of a powerful base, usually anhydrous ammonia, to instantly neutralize the broth. It turns what used to be a chaotic, reactive chore into this beautifully smooth, automated balancing act.
Moving right along to section 2, measuring microbial breath by tracking CO2.
So next up, we need to monitor the exhaust gases produced by our tiny biological workers. Which kind of sounds like a riddle, right? How exactly do you measure a microbe's breath? Well, water and carbon dioxide are the 2 most common end products of aerobic fermentations. And measuring that CO2 output is totally essential because it gives us the carbon balance and tells us exactly how fast the fermentation is moving along. But remember, we're talking about invisible carbon dioxide exhaled by billions of microscopic organisms mixed in a giant liquid broth. How on earth do engineers solve a seemingly impossible task like that?
This is where biochemical engineers step in with 3 brilliant solutions. First up is infrared absorption. CO2 actually absorbs infrared rays. So, by shining infrared light through the effluent gas and using a highly sensitive sensor, we can translate that light absorption directly into a precise measurement gauge. It's honestly a lot like using night vision to see the gas. Second, they can use a cool chemical reaction where gas bubbles through a solution of sodium hydroxide and phenol red. A photocell then measures the resulting color change to calculate the CO2. And third, they can measure the thermal conductivities of the different gases in the mixture. Now, while all 3 of these definitely work, that infrared night vision method, that's the absolute superstar of modern factories.
That brings us to section 3, the dissolved oxygen probes and keeping the factory breathing.
Because our aerobic microbes don't just exhale, they need to inhale too. Just like us, they require a constant, carefully measured supply of oxygen to survive and do their jobs. But here's the thing. Measuring dissolved oxygen inside a thick, complex fermentation broth used to be an absolute nightmare. Historically, scientists relied on chemical methods to determine biochemical oxygen demand, or BOD. The best known of these was the Winkler method. It was this super cumbersome process relying on iodine back titration. Just imagine trying to run a delicate chemical titration while interfering substances from a messy, thick biological broth are constantly skewing your results. It simply wasn't fast, and it definitely wasn't reliable enough for real-time factory control. Thankfully, modern engineering swooped in and gave us autoclavable dissolved oxygen probes that use some really clever electrochemistry. Generally, we rely on two main principles today: polographic and galvanic. Now, both use a special plastic membrane that lets oxygen gas slip through while blocking liquid and ions. In the polographic method, we actually apply an external voltage of 0.6 to 0.8 volts to a noble metal cathode, like platinum or gold, sitting in a neutral potassium chloride electrolyte. The oxygen diffuses through that membrane, reacts at the cathode, and the resulting electrical current tells us exactly how much oxygen is in there. The galvanic method is pretty similar, but get this, it doesn't even need an external power source. It uses a base metal anode, like zinc or lead, alongside a noble metal cathode. This pairing naturally generates its own electricity, basically acting like a battery powered by the dissolved oxygen itself. Both are just massive, game-changing upgrades from those old chemical titrations.
All right, moving to Section 4, the power of positive pressure and sealing the factory. So, now that we've tuned the liquid environment, let's zoom out and look at the physical atmosphere of the tank itself. We're spending all this time and energy perfectly balancing the pH, the carbon dioxide, and the oxygen for our specific microbes. But what happens if an uninvited guest crashes the party? We absolutely have to establish atmospheric control. So why is it so incredibly critical to maintain a positive pressure inside this massive steel vessel? The crucial point here is that positive pressure acts as an invisible, physical shield. We monitor this pressure super carefully using a device called a manometer. By keeping the pressure inside the fermenter slightly higher than the atmospheric pressure outside in the room, we make sure that air is always pushing outward through any tiny seals or valves. If that pressure were to drop for even a second, outside air, which is completely loaded with rogue bacteria, wild yeast, and competing contaminants, could get sucked straight into the tank, completely ruining the entire batch. So this outward push forcefully keeps the factory floor pure, while also helping force oxygen into the liquid for proper aeration. Pretty brilliant, right?
And finally, Section 5, the Master Control Room and Computer Automation.
We have reached the climax of our tour. We've looked at the hardware, the pH probes, the infrared CO2 sensors, the galvanic oxygen probes, and the manometers. Now, we finally meet the digital brain that oversees it all. But I've got to share a really surprising piece of history with you. Despite fermentation being a biochemical marvel, literally harnessing the power of living organisms, the fermentation industry, especially antibiotic manufacturing, actually lagged severely behind the general chemical industry when it came to adopting computers. It's true. Throughout the early 1970s, chemical plants were automating left and right, while a ton of fermentation plants were still relying heavily on manual observation and basic analog gauges. Thankfully, today, that situation is entirely different. Computer automation has completely transformed this chaotic biological soup into a precise manufacturing system. It eliminates manual intervention, drastically reduces human error, and automatically records every single data point for strict regulatory and safety requirements. Complex experimentation becomes an absolute breeze, too, because a scientist can just program exact shifts in temperature, pH, or nutrient addition and let the computer run the whole trial perfectly. Quality control is vastly simplified, but there's one more benefit that is truly, literally mind-blowing. As the source text outlines, in the event of a power failure, a properly computerized system has the ability to shut up itself and restart and gradually build up to the original level of activity. Just think about the sheer complexity of that for a second. A blackout hits, the mechanical stirrers stop, the aeration halts, everything goes dark. But the computer system instantly recognizes the emergency, triggers safe shutdown protocols to preserve the batch, waits for power to return, and then systematically resurrects the entire living ecosystem entirely through code. It carefully ramps the dissolved oxygen and nutrient feeds back up safely to avoid shocking the microbes. It's just incredible.
And that brings us to a final thought to leave you with today. We've seen how probes and sensors act as the eyes and ears of the fermenter, and how the computer acts as the brain. If advanced code and sterilizable hardware can effectively pause and successfully resurrect millions of living, breathing organisms after a total blackout, what other biological limits will computer-controlled engineering push past next? The microbial factory of today is an absolute marvel, but the biotechnology of tomorrow, it's going to be completely unrecognizable. Thank you so much for joining